Hello, I am Motalib Hossain, postdoctoral research fellow at Nanotechnology and Catalysis Research Center, University of Malaya. My research mainly focused on the authentication of food products using molecular-based or DNA-based techniques such as PCR, polymerase chain reaction, and DNA biosensor. Before starting my presentation, I would like to thank the organizing committee of 8th International Conference on Research in Life Science and Healthcare for giving me this opportunity to share my research at this online conference. I would like to extend my sincere thanks to all participants for sharing their valuable time with us. The aim of my presentation to share the advancement of molecular-based techniques in the authentication of food products. Outline of my presentation: I am going to start with introduction. Then I would like to discuss protein-based methods versus molecular-based methods. Next, I would like to discuss about various molecular-based methods with schematic diagram, and finally, recent innovation in molecular-based techniques. I would like to start my presentation with the answer of a question: why need food authentication? We know that among the other fundamental needs, food is an important and essential for all living beings because food plays an important role in the promotion of health as well as prevention of diseases. Therefore, we have to take food every day. However, all types of foods do not consume all group of people. Therefore, each group of people has their own choice in the consumption of food. Considering this fact, the authentication of food products is very essential and important. There are two main sources of foods. One is plant source, another is animal source. Among these two sources, the major concern about the food authenticity is related to animal sources because according to USDA, United States Agriculture Department, approximately 75% infectious diseases of humans come from animal origin. In addition, choice of food depends on various factors. One is the religious belief. For example, pork is totally forbidden for Muslim and Jewish. On the other hand, beef is totally restricted for Hindus. Similarly, vegetarians do not allow any animal materials in their diet. And some foods also have the sensitivity to specific person. After consuming this sensitive food, allergic reaction begins. Therefore, authentication of animal origin in food products is crucial. Moreover, adulteration of food products are rampant all over the world. For example, horse DNA was found in beef burger in UK. Chinese police seized approximately 20 tons fake beef, which was made chemically treated pork. Pork DNA was also found in halal branded Cadbury chocolate in Malaysia. Even pig DNA was found in fish product in Singapore. Considering this fact, nowadays consumers concerned about the authenticity of food products. Due to these circumstances, researchers have paid more attention to the development of authentic and reliable techniques for the authentication of food products. Mainly protein-based and molecular-based techniques are used for the authentication of food products. However, protein-based method is not suitable, particularly for the analysis of highly processed food products, because protein is heat unstable and denature easily. For this reason, protein-based method is suitable only for raw meats, and this method is less sensitive and it cannot differentiate closely related species and not suitable for multiplexing. On the other hand, molecular-based techniques use DNA molecule as a target biomarker, which is highly stable and specific and also sensitive. Molecular-based techniques are applicable for complex and severely processed food matrices where DNA is broken down, and it can differentiate closely related species. And molecular-based techniques also suitable for multiplexing. That means using these techniques, multiple species can be detected using a single reaction. For this reason, these researchers paid more attention for the development of molecular-based techniques. Among the molecular-based techniques, PCR, DNA barcoding, and DNA biosensors are mainly used for the authentication of food products. There are different types of PCR methods, such as in-point PCR or conventional PCR, PCR RFLP, and real-time PCR methods. Now I am going to discuss step by step all these methods with recent innovation. Now I am going to present about the basic principle of PCR techniques. Polymerase chain reaction or PCR is a technique which is used to amplify a single copy or a few copies of a segment of DNA for generating 1,000 to million copies of DNA sequence using a special enzyme called DNA polymerase. For this reason, PCR is called a copying machine for DNA molecules. Based on the number of targets, PCR can be divided into two groups. One is the single plex, which is able to detect only single species. Another is the multiplex PCR assay, which can identify the multiple species, more than two species, by a single reaction. Now I am going to present the schematic diagram of in-point PCR assay. To perform the PCR reaction, various types of PCR components are mixed in a reaction tubes. Among these PCR components, DNA template is extracted from food sample. Primers have to be designed and synthesized based on the targets, and other components such as tech DNA polymerase, deoxynucleotide diphosphate, magnesium chloride, and buffers are commercially available. After mixing these components, the PCR mixture is kept in thermal cycler, which is the PCR instrument. Due to the temperature gradients, the template DNA is amplified to million copies of the target fragments. This is the main principle of PCR reaction. Here I am going to present how we can identify the animal species in food products. Firstly, the DNA is extracted from food products using commercial extraction kit. The extracted DNA is mixed with PCR components, and after the PCR experiment, the extracted DNA is amplified into million copies of target fragments, and after amplification, the PCR products are visualized by gel electrophoresis. After gel electrophoresis, the PCR products are visualized on the gel as a band. Based on the size of the band, we can identify the species. Where single bands indicates the simplex PCR, that means presence of single species, whereas the two or more bands indicate the multiple species is present in the food sample. By this way, we can identify the individual species in the food sample. We develop a heptaplex PCR assay for the detection of 7 target species such as beef, buffalo, chicken, cat, dog, pork, and fish simultaneously by a single reaction. Where initially we developed a simplex PCR for the detection of single species, and step by step duplex PCR for the detection of 2 species, and triplex for 3 species, and finally heptaplex PCR for the detection of 7 species, where the 7 bands in a single length indicating the 7 species are present in the sample. And the electropherogram also generated 7 different peaks corresponding to these 7 bands, which also confirm the successful amplification of 7 target species by a single reaction. And this method was highly sensitive. The limit of detection was 0.01 nanogram DNA under pure state and 0.5% adulteration in atomic sample. This method is published in a ACS publication Journal of Agriculture and Food Chemistry, Human. We know that DNA molecule consists of many genes such as cytochrome B, ND5, D-loop, and so on. All existing PCR assay based on single gene targeted. For example, the cytochrome B gene targeted PCR assay, it is less reliable because if this single target is broken down due to food processing treatment, it gives the false negative results. To overcome this limitation, for the first time, we developed double gene targeted multiplex PCR assay where 2 different genes were targeted. One is cytochrome B and another is the ND5. This double gene targeted multiplex PCR assay has highly reliable and authentic techniques, and it has more advantages. For example, complementary targets is present, double layer security in this assay. There is less probability of false negative result, offering a confirmed detection. For the first time, we developed double gene targeted hexaplex PCR assay for the detection of beef, buffalo, and pork in the food products. Here is the 6 bands indicating the 2 targets for beef, 2 targets for buffalo and 2 targets for pork. Uh, where electropherogram also generated 6 different peaks corresponding to 6 bands. This also confirmed the successful amplification of double gene calculated multiplex PCR assay for the detection of beef, buffalo, and pork. The sensitivity of this method was 0.02 nanogram under pure state and 0.1% adulteration under ethnic mixture. This work also published in Q1 Food Control. Next, I am going to present PCR RFLP. PCR restriction fragment length polymorphism or PCR RFLP is also PCR based techniques where PCR products are digested with a special enzyme called restriction enzyme. After digestion of the PCR products, the generated restriction fragments are analyzed to identify the diseases in food products. RFLP is the most popular approach for analysis of genetic variation. The schematic diagram of PCR RFLP techniques. The restriction enzyme and buffer are mixed with PCR products. Then this mixture is kept in a water bath for digestion of the PCR products. After digestion, the electrophoresis is carried out to visualize the restriction fragments. In the gel image, the restriction fragments are clearly visualized where 210 bp PCR products, after digestion, it generated 2 different restriction fragments, whereas 260 bp PCR products generated 3 different restriction fragments. By analysis these restriction products or restriction fragments, we can detect or identify the species. Whereas the mechanism of the restriction enzyme, after bind, the restriction enzyme is bind to the specific site of the double-stranded DNA, and after binding the specific site, it can generate 2 or more fragments. This is our developed PCR RFLP techniques where we digested 90 bp PCR products with restriction enzyme and generated 50 bp and 40 bp restriction fragments, whereas when digested 106 bp PCR products, we generated 87 and 90 bp PCR products, and so on. This method is also published in Q1. Next, I am going to present the DNA barcoding technique. DNA barcoding method is also PCR based method where an additional step is involved, which is called the sequencing. In DNA barcoding techniques, cytochrome oxidase 1 gene targeted biomarker is used, which amplify approximately 650 bp fragments through the PCR reaction. To detect the diseases using the DNA barcoding techniques, firstly DNA is extracted from food sample, then the extracted DNA is amplified using the PCR techniques. Next, PCR products are purified using the purification kit, and PCR products are sequenced using the sequencer. And finally, the PCR product sequence are compared with the blast search tool in GenBank or board identification engine. And from the comparison with the gene bank library and matching with the BCR's sequences, we can identify or detect the species using the DNA barcoding. Next, real-time PCR assay. The conventional PCR method only gives qualitative results. How much adulterants are present in the sample cannot quantify using the conventional PCR methods, which I discussed previously. On the other hand, real-time PCR assay gives both qualitative and quantitative results simultaneously. For this reason, real-time PCR is also called quantitative or QPC. The real-time PCR assay is a specialized technique that allows a PCR reaction to be visualized in real time as the reaction progresses using fluorescent dye. In real-time PCR assay, no need post-PCR processing like electrophoresis, which is needed in conventional PCR assay. We also developed real-time PCR assay for the quantification of beef, buffalo, and pork DNA in food sample. The method is able to detect up to 0.003 nanogram DNA in pure state and 0.1% adulterant in food sample. Our another real-time PCR assay for the detection of cat, rabbit, rat, and squirrel DNA in food products. Now I am going to present my last technique, DNA biosensor. Although PCR is popular and widely used methods for the authentication of food products, however, this method has some disadvantages, like it is lab-based methods and needs highly sophisticated instrument, and it also requires very long time to perform the experiment, and it needs trained personnel to conduct the test. To overcome this limitation, nowadays researchers pay more attention to develop the DNA biosensor, because DNA biosensor is the rapid, less expensive, and field-based methods. The DNA biosensor is comprised of a biological recognition element and a transducer that can detect the particular biological reaction and converting it into a signal. The components of DNA biosensors include transducer, bioreceptors, analytes, and detectors. Here I would like to present the schematic diagram of DNA biosensor. In DNA biosensor, transducer is used as a device which can convert the one form of energy into another. Firstly, these transducers are modified with different types of nanoparticles. Here I am going to present the graphene oxide and gold nanoparticle based DNA biosensor. Initially, the transducers are modified with graphene oxide, and after modification with graphene oxide, it also modified with gold nanoparticles. This modification gives the two advantages. One, it increases the signaling power of the transducer, and second, it facilitates to immobilize the single-stranded probe to the transducer device. And after immobilization of the probe, it is generated or it works as a electrode, and when we add the target DNA after extraction from the food products, this target DNA hybridize with the single-stranded probe DNA due to the complementary sequence. After hybridization, it gives a signal. By this way, we can detect the species in the food products using the DNA biosensor. Thank you for your kind